hi to everybody. So we are really pleased to see here in Moscow, in Abtec office here, Mr. Дэвид Андерсон from Aber Instruments. And we will have just a short discussion and Q&A session maybe to listen about how to work in such company which is related to the really cutting-edge technologies and bioprocess equipment, and how it grows, how it started, and what is to work in such a company. What qualities personal you should have to drive this. Yeah. So, Dave, maybe you will say a few words about your company first of all.
Sure. Well, Aber Instruments is the market leader in online biomass monitoring, and as a company, it was originally spun out from Aberystwyth University in Wales 25 years ago. Last year we celebrated our 25th anniversary. We've been through a classic growth curve of a small and medium-sized enterprise in those 25 years. I've been with the company nearly 2 years, and we have a fantastic array of modern product, ready to help the scientist and the online bioprocess technologist.
Yeah. Okay, good. And what is your background actually? How did you started into this?
Well, I took my first degree in applied biology, and then I went to work at Cardiff University for 5 years. Made the classic transition from bench research scientist into sales. Found that I liked it very much. I worked with a tissue culture company and it sold immunochemistry products and laminar airflow devices for 3 years. And then I moved to Pharmacy at Biotech, and then of course that was bought by Amersham Life Sciences, and we became Amersham Pharmacy at Biotech, and then eventually GE Healthcare, and I rose to be a senior specialist in protein chromatography, working at lab scale to process scale devices. I enjoyed it very much, but for personal reasons I left, and I joined Aber 2 years ago, and as a European sales manager. And it's been a fantastic experience. Great job.
Cool. Sounds cool also. Okay, and could you also explain a little bit how this technology, based first of all on normal physics, I would say, how it went to the biosciences,
Right.
And of course, we see now the tendency to go to the cell culture in regard of bioprocess.
Sure.
But if you are so old company, let's say it's 25 years, it's quite sensible.
Mature.
So, So how it moves and how this is developed exactly from the normal physics to this.
Sure. Okay. Well, one of our founding directors was Professor Douglas Kell, who recently retired as chairman of the Biotechnology and Biosciences Research Council in the UK. When he started off as a young lecturer at Aber University, he decided that he would like to be able to monitor concentrations of bacteria in wastewater. And at that time there was what was called the Centre for Alternative Technology, fairly close to Aberystwyth, who had a lot of expertise in perhaps ecological technology. So he went to CAT and said, this is what I want to do. And one of our other founding directors, Dr. Bob Todd, was there. He's an electronics genius. And he realised that if you have a cell with an intact membrane, it can express capacitance if it's put into an electric field. And so he created the field of what we call dielectric spectroscopy. And over the years, as the technology has improved and modern electronics have become more sensitive, we've gone through 3 generations of machine or system to end up with the Futura for biotech and the compact for brewing applications that we now have. How does it work? Well, if a cell with an intact cell membrane enters an electric field, the cell membrane becomes polarized, and the cell exhibits capacitance and the ability to store charge. And we can measure very fine phase angles between the capacitance of the bacteria and its surrounding liquid culture media. So in a way, the analogy that I use is that if you rolled out all the intact cell membrane like a sheet, the total surface area of intact cell membrane is proportional to the viable biomass in the sample, because it has to have an intact cell membrane to be able to express the ability to store charge. And that's the root of what we do, and that's how the technology has evolved.
Yeah. Sounds very, very interesting. Yeah, we will know a little bit more about your technology. And also another question is how fast it was the market response for the technology you have, this platform, and how hard is actually to drive this kind of business. I mean, not when it's already developed, but really from the early development stages to come to the really concrete, ready product. Yeah?
Sure. Yeah, that's a really interesting question, and I'm not sure I'm the best person to ask, seeing as I've been with the company only a relatively short time. But initially a lot of systems that were produced, one classic one which is still in fairly common use is called the VCM 520. That was an early device. It looks like very 1980s technology. Yep. It's large, it's clunky, it's got switches, and now as time has moved on and we've become established with all of the leading biopharmaceutical companies globally, and with our yeast monitoring products in many of the major breweries as well, the size of the unit has shrunk. We're always responsive to customers giving us new ideas, suggesting improvements. Probably the biggest advance that we've made is in reducing the size of the equipment, increasing its sensitivity, learning how to use software to manage data and take raw data and present it in a usable and understandable form to the scientist. The other issue, of course, is that the demands of the biopharmaceutical industry has become very great in terms of validation requirements and GMP, and we've worked very hard to develop an IOQ package to fit with the needs of the research scientist.
Yeah. And the other question is also the cooperation, the collaboration, let's say, with the general science centers in worldwide or in the UK.
Yeah.
How strong is this? How big is this? And could you tell something more about this?
Of course I can. It's very strong. Like I've said, many of our customers are leading pharmaceutical companies and specialising in biopharmaceutical manufacture. And we're always taking ideas from them to help us develop new product. I can't really say too much about product that we have in development, but we have a constant pipeline and a fairly large R&D team who are constantly working to try and bring new product to market with us. And that is led by customer idea. And we have a very active stream, set of local distributors as well globally. And our distributors are very good. We select them very carefully, and we're always prepared to listen to their feedback, give their ideas on product development and new applications. So some are very, very open and very good and very sharing in what they give us, and we take that information and move it forward through the production pipeline.
Yeah. And also another one is, for example, in Russia, we also have some scientists who want to trying to develop the product and to bring it to the market, based on some science ideas they have. But to be honest, at the moment we really don't see a remarkable or something significant science that is close to be a really market product, especially worldwide
Yeah.
product. Yeah, and why we are trying at the moment to understand and to improve it, and what do you think is the key factors to be able to connect the science with the industry or with the really profitable business with the market?
Okay.
What do you think is the key factors on this?
Well, again, it's a really interesting question. I think finding the idea, having a good personal rapport with your scientific base, your scientific advisor, stroke inventor, if you like. Having access to funding is a major issue as well. So, but you know, money can be put behind these ideas, and often making that crucial triangle between idea and commercial exploitation and funding is really the way forward. Aber has probably taken a slightly different model to that and has sort of developed many of its products itself from, you know, inventor suggestion.
Coming from academia?
Yeah, coming from academia, but also taking ideas from industry and modifying them. I think you have to be very close to your market as well and understand, you know, how a new invention can be taken and applied and perhaps refined to fit your particular market niche better. So yeah, those are the key factors. I mean, often, like so many things, it boils down to personal relationship and a good contact between the commercial developer and the original inventor.
And also it's the relations in this triangle, I mean relations between university and academia. I mean, there is some basic knowledge and science is very tight, and so you need to have the science centers, etc., etc. And working in a very free form of knowledge exchange.
Absolutely. In English or in Britain, we've had a lot of success with incubator style businesses where small academic spin-offs have been entered into a local sort of building or research centre where those relationships are developed very well. Another sort of important factor, once an incubator building is working well, or an incubator centre is working well, to expand that into a cluster. And I know the Russian government is very keen to develop biotech clusters at quite a few sites across the country, particularly here in Moscow and in Kaluga. So, you know, there's a lot of interest, and I would say that your government is doing all the right things to sort of push the biotech business forward.
Okay, very politically right answers.
Well,
okay,
I do my best.
Yes, I think that's very nice information that you shared with us, and your ideas, your thinking on the situation, and your answers is very. Thank you for this, and it was very nice meeting you here and talk to you. And I hope this information, these few words, will be also somehow interesting to the guys, to the people from the both sides, or from industry and from the applicational processes specialists. Yeah, so thank you very much.
Thank you for inviting me.
And
thank you, Michael.
Yes, thank you too. And come back again to Russia.
I certainly will. It's been a very enjoyable trip so far.
So, Abtec TV. Bye-bye.
Thank you.
Yeah.